So good afternoon, everyone. Welcome to this edition of the NITheCS Colloquium. My name is Francesco Petruccione, and I'm at the moment the interim director of NITheCS. Yeah? This afternoon, we have the big pleasure of having again as a speaker Professor Erik Aurell. Yeah, and Erik, just as a reminder, in case you missed his previous talk, studied at the University of Göteborg, where he graduated with his PhD in a few moons ago. Yeah, but since 2003, he's a professor of biological physics at the KTH Royal Institute of Technology in Sweden. So if you are planning to get a Nobel Prize, you should be good friends with Erik. Yeah. Over the years, he was a guest professor at various universities in Finland and in China. And today he will speak. Last time he was with us, he spoke about his quantum interests. Yeah, but he's a biological physicist at heart. So today we asked him to speak about his more recent work on COVID-19. Yeah. So Erik, we are very, very happy that you accepted to be with us again this afternoon, and we are really looking forward to your talk. Yeah. And maybe while you share your screen, I will briefly introduce the other people that you see on the panel. Yeah. There is Professor Martin Bucher, who is a NITheCS associate. And he's the PI of the NITheCS COVID genomics project. Yeah, so and Martin kindly offered to moderate the questions at the end of Erik's talk. You see also Professor Ying-Se Ma from UKZN, who is also a NITheCS associate and a long-term friend of Erik. So he kindly joined us as well, and I'm sure he will also assist Martin in the moderation of the questions. And as usual, there is Professor Sinaisky, who is the canonical co-host of these events, and he will give us also all his support. Yeah. So Erik, I think I don't want to speak any longer. If you would like to share the screen, you're most than welcome to start with your presentation.
Thank you very much.
And maybe the usual reminder, sorry, to the participant to use the Q&A facility to ask questions. Sorry, yeah. Erik.
Thanks. It's a great pleasure to talk to you again, and everybody. I can see there are now 26 participants, and I checked while the introduction was going on that at least one of my co-authors, Professor Ongli Zheng from Nanjing University of Telecommunications, is in attendance. So also some of my co-authors joined. I'm not sure the others have joined yet. I didn't have time to check. So I realize I'm this time speaking to experts, not that last time. I'm just joking. So, but I think it still is an interesting kind of excursion that of simple and less simple data analysis that you can do on an unprecedented data source available to scientists with all backgrounds. So let me start by sharing my screen, and then I will introduce the topic and what we have done as we go on. Okay. So let me go to the first slide. I hope you can see it. You see it well?
Yes. Yeah. It's clearly seen. Yeah.
Very good. Okay, very good. And of course, I am happy to take questions at any time. I know they are moderated in this for this colloquium, but I have no problem. I mean, of course, I welcome questions or remarks, or both during the talk and after the talk. So I am going to speak about two projects. There's two papers, and then various follow-ups which we haven't written up in the form of formal papers, but the references to the two papers will come later in due course. Let me start by defining for you GISAID, which is the Global Initiative for Sharing Avian Influenza Data. I hope you can see it right. For me, the chat and so on the top of the screen. Can you see the whole screen with the text and so on? Or maybe it's a Zoom issue.
Yes.
Yeah, it's okay. So
yeah,
so
yeah,
Of course, GISAID was started with this sharing avian influenza data. Sometimes you can see this is the original name. Sometimes it's called also all influenza data. But I would say in the last year and a half, it's become mostly famous as the leading repository for SARS-CoV-2 genomic data, which of course is not strictly speaking influenza and neither so avian influenza. It's run by a voluntary association in Germany, and I think it's mainly supported by the Federal Republic of Germany. And it holds, as of when I checked last time, it was on Friday, almost 3 million submissions about SARS-CoV-2, the virus, which of course, you know, everybody knows is behind the current pandemic of COVID-19. So it depends what you want to use the data for. Not all of them are high quality full-length genomes, which is the only kind of data which I'm using here, but a very large fraction of this data contains all the genomes of the whole genomes of the virus which have infected different patients at different times. Okay, and this almost 3 million such data with a lot of metadata available, such as what is the lab that did the sequencing, what is the date when it was sequenced, what is the date when the sample was taken, where was it taken, and sometimes even other data. So it is for a data science point of view, which I think is also in the ambit of the scope of work of your center in South Africa. This is actually an unprecedented data set to try out any kind of data analysis that you like to do. And of course, it's also a societal problem of the highest importance. This pandemic has changed the world more than anything I have seen basically in my lifetime. And I'm not so young anymore, as Francesco pointed out very politely in his introduction. So I suppose that at least some of you listening to this have maybe looked at GISAID, maybe looked a lot at GISAID. Some of you maybe haven't looked at it at all. And some of you may not even have heard the abbreviation GISAID before. So I pitched my talk mainly to the last two. So if you've never heard of it before, or if you heard of it but never did anything, or you didn't try to do anything, then this is a talk for you to raise your interest in this data source and what can be done and what so far has not been done. Some simple things. I will start with the simple things, and then I will tell you about some things which are a little bit less simple from the data science point of view later. Okay, so here is a very nice quote from a paper which I found when preparing this talk, which was published in Nature Reviews Microbiology this year. And it basically says in very nice words that the resource, GISAID as a resource, which allows you to study local outbreaks and inform public health policy, is a new age in virus genomic investigations. So since a lot of the infectious diseases which afflict mankind are viral in nature, I think it's fair to say that this is the dawn of a new age in understanding, monitoring, and using massive data on pathogens of all kinds. It's only more or less a couple of orders of magnitude of difficulty in assembling the genomic information from the virus to bacterial pathogens, if you like to do that also. Now, the very first thing you can do is to go into their own website and just check what you see. And what you can see on this kind of picture that you have down there. This is already quite an old one. It's from more than a year ago. And you see some phylogenies. This means inferred relations of inheritance of the different viral strains. And then plotted on top of those, you see mutations, not so many, in fact, here. And these mutations are plotted on top of these, on top of this tree. And now, what I want to say about showing this picture, which is already more than a year old and taken from their own website, is that When you start to the left, it looks fairly clean, and you can discern by eye some things. But the further you go to the right, the more it looks like a data cloud. And the only thing you can see are very, very gross features, like it's more red in the top, whatever red actually is here. And it's more red in the very bottom. And it's kind of green, yellow in the middle. And the tree itself has so many branches, you cannot discern them by eye. So there is other things that you can do except plotting a data cloud. When you have lots of data, big data, you have to find other things to look at. And this talk is about two things that you can do among the many, many other things that you can do with this data set, this kind of data set. So I would say two stories. One is this rather simple time series analysis, which you can find on bioRxiv, the biological preprint archive from April. I will also show some more recent data, so up to July this year, but basically with the same idea. So at that time, when we wrote up this paper, there were 500,000 whole genome sequences on GISAID. Now, of course, today, in end of August 2021, there is many, many more than 2 million. And then a more ambitious and speculative theory and the data analysis that go along with it. So it's more like a glimpse of other things that you can do, which we did already in the first year of COVID, and it was published in PNAS last December. So the paper, the reference is is published in early December 2020. And also I will give some more data which are a little bit later, but the data for the second part of the talk will be basically using up to the end of 2020. And it's ongoing to check this up to today, but that will be in a follow-up talk at some point, or when the next papers will appear. So I will start with the simple things. After telling the necessary background of the virus, coronaviruses is a family of families. There's the subfamily to which the SARS-CoV-2 belongs, is called more technically the beta coronavirus family. And I think everybody has seen these pictures in the news. You know that there is genetic material in the form of RNA, which is inside a capsid, inside kind of a container. And then the outside container, you have spikes going out. Those spikes are for in the protein, also called spike. The RNA inside codes for about 30 proteins in a rather peculiar way. This is a very nice illustration, which I pilfered from a very nicely done illustration from a paper from last year. But basically, you can find many versions of them online. There is a long sequence of co-expressed genes, which are known as the ORF1AB. This means open reading frame 1 in part AB, because there's a frame shift in between. And this is translated as one long polyprotein, which is then cut into separate proteins which have separate functions. And those ones in this ORFAB, which is about 2/3 of the whole genome, so about 20,000 base pairs, or a bit more. They are divided up into proteins which names as NSP1, NSP2, NSP3, and so on. And they stand for non-structural protein 1, 2, 3. These are proteins which do things which are important for the viruses, like the helicase, which unwinds part of the stuff, and other ones. Then comes the spike protein. So that's translated as one unit. And there is since this is of great interest, there is in the lower part of the screen, there is an enlargement of spike with the well-known receptor binding protein, where it binds to its target, the ACE2 receptor, and certain known places. Here there is a 483. This is 614, which will appear again. And the terminology, when it's written D614G, it's that the wild type, that means, in this case, the first sequenced version of the virus, which was sequenced in early 2020, and usually goes by the name of Hu1 or sometimes Wuhan, because it was Hu and Al who sequenced it, and it comes from Wuhan in China. So it has the letter D, an amino acid with the code D, in position 614 in this protein in the wild type, and there's a mutation which tended to G instead. So that's an important one. It will come up again. And here's another one which comes towards the end. So you can see that this is a rather large protein of something like 12, almost 1,300 amino acids, so correspondingly larger in terms of RNA. And then comes all the rest of the ones. So ORF3A will appear again. So will ORF8. So will M. So will N. And there are E and others which are inside here, color coding. Of course, the scale is such that they cannot be discerned. Okay. Yes, and for this, I'm now going to tell you about 3 natural experiments with this data, namely the growth of the variants of concern, which have been very much in the news for quite some time. And I will just skip directly to the next slide. This is, let me see, so Hongli Zheng, I guess, is in the audience, will also be able. Yue Liu is her young student. Kajsa Torell is collaborating in both papers. She's now at the University of Gothenburg, my alma mater in Göteborg. Rikard Nordén is also there in the department. These are both in the Department of Infectious Diseases, and these two are in Nanjing. So you can see basically most of the material which I will tell you is in the paper which is on bioRxiv, but I will also supplement it with some more, a little bit more recent data. So those variants of concern, those are the ones which more recently are called alpha and delta and so on, and we'll do them in order. So the UK variant, as it used to be called, but that's not supposed to be politically correct anymore. One is supposed to call it alpha, but I suppose many of you know it as the UK variant. It has another more numerical or formal definition in a certain classification of variants, which is called then the B117 variant. And I'm going to tell you about the difficulty in finding out what is actually this variant. And we looked and looked and looked, and after a while we found that the first definition is in a technical review from Public Health England, which was published in December 2020. So it says 2020-1201. Of course, that's not the first detection of this one, but that's the first publication from Public Health England on this variant of concern, when it was raised to a variant of concern. And if you read this report, which is now a bit old, you can see a list in a form of a table of which are the mutations, which I list here both in the nucleotide format and the amino acid format. And so here, these are all non-synonymous mutations, which means that they all change the amino acids. They start in this long ORF1AB with the first one, which is the mutation C3267, which goes to a T in nucleotides. And here's the amino acid. I think this must be counted from beginning of ORF1AB as one continuous polypeptide. Okay, and then there's a lot of them in spike, and then there's ORF8, and then there's also N. Okay, so there's a list, there's 17 of them. Then there's also in the above the same table, there is a list in text where there are 6 synonymous mutations. So these are mutations which do not change the amino acid. Therefore, there's only 2 columns here. There's a gene name, which starts with ORF1AB, And the first one here is the 913 nucleotide, which is changed from C to T. And it ends with an M. So M is one of the last, towards the end of the genome. So 26,801C. I should comment that this one, the spike mutation D614G, is not listed in this document that I'm referring to from Public Health England, but it is referred to, it's listed in other definitions of the alpha or UK variants. You might otherwise also find D614G, but here we are following the original one, so it's not there. And okay, so now we have this data up to today, almost, well, maybe more than 2 million in any case, genomes. We have the data when they are taken. We can compute frequencies of all these mutations, and we can stratify that by month. So you get the time series on how do the different mutations or the wild type at these 23 positions, how do they change? Do they change together or do they change a bit randomly? That's the first question, okay? And so here's what one finds. One plots them, and one finds that there are actually two of them which are really different. One is the one which is pointed to by the red arrow, which is one of the synonymous mutations, 26,801. But there's also one more, which probably, that is our guess at least, was mislabeled in the report from Public Health England, because it has exactly the opposite. What you see here is the frequency of the wild type. That means the original one, okay? So as this variant grows, the frequency of the wild type goes down. That's why you see in early 2021, you can see that most of these frequencies, they go down together, but there is two outliers, one shown and one not shown, because it goes precisely in the opposite direction as the bunch of the frequencies. And here you find that was up to earlier. Now here is all of them up to except the deletions, and that's up to July this year. And the time series shows you different things. Okay, now the ones that we have, I excluded deletions here, and I've also excluded the one which was on the previous slide, 26,801. You can see that there are two outliers here. Okay, clear one. One is the 681 mutation. That means what's plotted here is the wild type, and you can see that the wild type, this is the yellow one, which eventually goes down to, if you can see the cursor here, it eventually in wild type goes down to 0. That means everybody is a mutant here, while in many of them the wild type goes up. So here there are not many mutants left. So this one, which was in the original definition, it has a different dynamics. It goes to fixation, while these ones here, they go back, okay? So it must mean, of course, that this one, if it went to become such a high prevalence, the mutant here becomes so prevalent, it cannot be due to growing together with all the other mutations in UK and the UK variant, because they go up here instead. Then there is also here, there's some variability. You can see that there is the N501Y mutation, which is has the wild type here has less frequency than the rest of the guys. So there's some structure also inside the ones that have more or less the same dynamics. And this can be seen more precisely or more accurately by looking at the frequencies of now the mutations in this list as a function of position. So if they would all grow as one big clone, you would see that this thing would grow as lines which come up and go down together. That's not such a bad description, but there are certainly, and you can see it clearly by eye, there are exceptions. So there's this one at 11,288, which is a deletion, which peaks a little bit earlier than the other ones. It seems, at least it's not on the same frequency as these ones here. So you see, here is where they reach in February 21, this UK variant reached its highest one, and it went to almost 50% in the beginning of the genome. Here you have 4. I think you should disregard the first one here. 4 here, and then this one is a bit lower. That's a deletion. Here also there's a deletion, and here there's other stuff happening inside spike. So there is an initial tendency for all these mutations to grow in synchrony, but later there are some variations which appear along the genome here. They're not really growing together completely as one clone. Now I go to the South African variant, and of course I realize that you are the main experts of this, and you also are the co-authors of the original definition. So we have not followed your paper. We have followed to be following up from the same source as the other one. There is a later technical briefing 6 from Public Health England, which we have followed here. So with apologies if the English later gave a different definition than you did, but this is what you find for the South African variant when you do the same kind of analysis. Here, of course, here's the table. And there were only non-synonymous mutations listed by Public Health England, 17 of them. And only 7 of these were only 7 retained in this kind of classification with letters and numbers, which is called the pangolin classification. And those are the ones which are marked by green. That's 5 in spike and 2 outside spike. And the overlap with the UK variant, which was before, I have marked with blue here from the right. So that's this one, N501Y, and the deletion here at 11,288, and a few more. Okay. So when you plot them like this, and here I have not plotted, we have not plotted deletions. There's only one overlap with the UK variant, and this is the dynamics of the UK variant. So here is the one you have basically seen it already. This is the wild type frequency of the N500Y. Now the rest of them, of course, in this plot here, because the South Africa variant never reached as high prevalence worldwide, it didn't go to almost 50%, which means that the wild type on these positions never went down to 50%. So it looks in the scale as there is maybe some variation, maybe not so much, who knows, here. And it's more interesting to look at the blow-up. So we're going to look only at the frequencies of the mutations along spike. And you find the following situation. So there is one which went up here. That's the first one listed in the Public Health England definition, which is spike D80A. And that went up at some point to almost 10% worldwide. There is another one, which is this E484K, which also went up to 10% or something, but they didn't go up to the maximum at the same month. You can see that the color of this one and that one are not the same. So they peaked at different times. Okay, so this peaked in April 21, and this one here peaked a little later. Then there are others. Okay, this one seems to grow, this one towards the end. The last one listed in spike grows later. And there are some of them, actually, there are 2 of them right next to each other here that grow very little. So they've only grown up to maximum 2% worldwide. That's these ones over here. So there is quite some, there's at least a factor 5 in prevalence reached here. Okay, sorry for the going a little bit forward. Okay, but I was more or less done. So then I go, next one is the India variant, which is the one which is most prevalent now, which has the pangolin name B117 2. It's also supposed to be called Delta now. You can find definitions in NextGen and in pangolin, but we haven't found any list from Public Health England in their technical briefings, but there is a GitHub page pointed to in the technical briefings, which in fact coincides with the pangolin list. So there is an overlap there. And here it looks like this. So next strain, which is this data source built on top of GISAID, has 18 synonymous mutations, and it calls this 21A delta. Those are the ones listed here. And 6 of them, which are written here, which are marked by red, are not in the pangolin definition, and the missing ones are marked with red arrows from the left. And then I marked for convenience 2 of them. One is that this both next strain, but not pangolin, includes the D614G mutation in the list. And so that's marked by black here. And as you can see, there is a reason for that, because it was a very high prevalence already when the data gathering started. And the other one is the one which is common with the UK variant, which is in this case spike mutation P681R. So it's the same position which is mutated, but it's not mutated to the same. No, there's a difference here in the 2 of them. So the wild type is the same, but the mutations are to 2 different places. Okay. And here is what it looks like here. What you can see is that also for the India variant, there is 1 mutation in N, which really has a different dynamics. That goes, this is of the wild type. The wild type goes down, goes up again. So it must appear in other combinations also. There is the one which is 681, where the wild type goes down. We have seen that curve before. And there's yet another one, which is the spike mutation in spike, which is R158G, which starts at the reasonable level of the first month where we have data, which is March 2020, and then it goes down very quickly and have stayed down basically since. So also here for the latest one, it's not a question that these kind of overlapping or partially conflicting definitions is something that was only in the beginning, but also India, the India variant, which has become a concern only now this year. Also here we can find different dynamics of the listed ones. And if you now go only to look at the across the mutations in spike, you can see the D614G, which started out already at something like 70% and has now basically reached fixation, so that almost all samples in the latest months have this one. There are the other ones which don't, which are around 90% now in spike, and which went up, started. So I'm talking about this one, that one, that one, that one, and that one. Although they are not exactly the same, there is a difference in some percent in the frequency worldwide. And there's one which hasn't grown at all. This is this R158G, which was already on the previous slide. So what one learns about this, the observations, is a simple time series analysis where one plots frequencies of mutations at different points. Of course, it was impossible to do, especially for an amateur before GISAID. I haven't seen it done very much. And it does give information which is immediate with visual inspection. That means you have some frequencies of some things are definitely following a different dynamics. They go down when the others are not going down and so on. So this is qualitative information which is easy to get and which is very convincing when you see it. It shows that some listed mutations must be wrong. This is not really for that means wrongly classified. They cannot have been part of the same cluster of cases growing together. This is not for criticizing anybody else. It's for saying that when you are looking at this from the outside, not only can it be a little bit difficult to find correct definitions of all the data sources, also the data sources in the haste of the pandemic have sometimes surely been made up quite quickly, and there may be reason to go back and check that everything is what it seems to be. Now, the rest of the ones, even filtering out all the ones that are obviously different, the rest do not seem to grow completely evenly. So either there's a mixture of clones or there are other effects going on. And with this, I think I'm through my first half, and I have to check how many minutes do I have left for the second half, which is the more. Maybe the chairman can tell me.
Erik, you have about 20 minutes or so. Yeah, but you know, give or take 5 minutes, nobody will die.
Yeah, very good, very good. No, I think this is the simple story, and I hope I have inspired others to go on and do. Maybe I should say something about the morals. I think the moral is that you can get information that means really something that you can understand and know what it means from this data by doing simple analysis, which kind of can be masked if you do massive computer-aided data analysis. I think it perhaps speaks more on our ability of human to take into information. It's not only the data clouds which obscures vision, but also other ways of plotting the data and classifying the data. If you do it with automatic support, can mix things, mask things, which is good for our understanding. So with the end of this story, I will move over to the other story, which was mainly done a year ago, and the paper was submitted in the final form almost exactly a year ago, and then published some months later. And it's kind of connected, though. It's to say, can there be things which are, and I'm not claiming that there definitely has to be things of the type which I will soon talk about, but there could be. And at least the data shown now on these 3 experiments do not give an ambiguous reflections of clones that grow separately and well separated from everybody else. Okay, so maybe there is something here more than what so often other does. This, of course, goes to phylogenies and perhaps to confounding factors in phylogenies. So I call this part of it Reflections of Viral Recombination, but there is a paper title, which is Global Analysis of More than 50,000 SARS-CoV-2 Genomes Reveals Epistasis Between 8 Viral Genes. That's the biological sounding title which we had to make to get into PNAS. 50,000 because this was done on data available on GISAID until August 2020, so a year ago. And I will show you some data which were done until December 2020, so with more data, but we are still ongoing the analysis until today with the more than 1 million data today. So let me go very biological, but very briefly in 4 or 5 slides. So to start with, what are the forces of evolution? Well, the first one is natural selection. And here you have one of the most famous examples, 4 pictures of Darwin's finches. The next one is mutations. And mutations I've already talked about in the context of the data which we have available on SARS-CoV-2 on this side. Here's another illustration showing genomes or parts of genomes, in this case actually amino acid sequences of proteins in a protein family. And those sequences are much more diverged because the timescales here are much longer than the ones I talked about before, but the idea is anyway similar. You can see there's some conservation. There are some columns where not so much change, and there are other columns where a lot of things change and you don't see anything basically. There are some deletions, precise as you can also see. You could also see in the other data, and those are written here by these dashed lines. So it's just an illustration. Well, there's also a third, but there are more. But for this story, there's the third one, which is recombination. And so recombination is, of course, what we have in human genetics and in all higher genetics of all higher organisms. In all mammals, for instance, is that you have a mixture of genetic material between two kinds of two classes of organisms, in human called male and female, which give rise to the next generation. There is, according to a well-known hypothesis, you have lots of recombination between organisms in the same species in higher organisms. According to a well-known hypothesis by Bucher, it was common between all organisms in early life on Earth. In bacteria, it depends. Some bacteria have a lot of recombination, some have very little. Some of them have variable. It depends on their on where how they're living. And in viruses, it's also different. But in particular, it's well known and has been known for a long time that coronaviruses, due to their mode of DNA replication, have, at least compared to other viruses, quite a lot of recombination. Recombination is an ongoing fact in coronaviruses. And if you Google it, you will find many articles arguing for the origin of the SARS-CoV-2 and other more recent coronaviruses due to recombination from parent strains, which then could have been from different hosts, so bats or whatever you have. But there is also a number of reports on ongoing recombination between strains that co-infect one patient in the ongoing pandemic. I just listed three here. I think basically because the first and the third are from Cell and Nature, so of course one has to give those. Maybe the second one from Journal of Infectious Diseases is the most interesting for our purposes. There are more reports in this direction. So it definitely is so that the SARS-CoV-2 exhibits recombination in the lab. There are also other reports in this direction, and recombination in one patient. The question is, of course, how much? And I will assume that it is quite significant, that it is not an effect that can be ignored. But that I would say that I have to caution is a hypothesis, and then which I'm going to use. It's not something that's proven. It's just proven experimentally that the effect exists. Okay. So now, what does it happen? What is the important, let's say, from the viewpoint of evolution and evolving population? What's different of having sex and not having sex? What's different between bacteria that they evolve without sex, which many of them don't do, or plants and so on, and animals that have to have it, and then also viruses that may have it in a significant amount. Well, there is a theory developed by the famous geneticist Kimura from Japan, Kimura, in the 1960s, which connects this to an even earlier and very famous theory from the beginning of the last century, saying that if you have a lot of recombination, and I'm not saying that that definitely has to be a lot in SARS-CoV-2, but at least there is some. So if you have a lot of recombination, you will have a stationary phase of the distribution of genotypes in a population, which can be seen as a small perturbation away from the state usually called linkage equilibrium. Linkage equilibrium is the phase when recombination is so fast that the genetic variants, in biology terms called alleles, at different loci, and that's biology terms for positions, are independent. So you can have the frequencies on one locus is independent from the various, what you find in one locus is independent from what you find in another locus, and either they could be all the same, or they could maybe be half-half of two different variants, but you don't have any correlations between these two. That's linkage equilibrium. In quasi-linkage equilibrium, small correlations remain because recombination, though assumed to be a fast process, is not infinitely faster compared to the other ones. So the effects due to in the phase of quasi-linkage equilibrium in a population evolving under natural selection, mutation, and recombination, where recombination is the fastest process, is the was discovered by Kimura, and there's this beautiful two papers by Richard Neher, who is also the main maintainer and the main person behind NextStrain, and Boris Schreiman from around 10 years ago. And we wrote a paper together with my long-standing my colleagues since way back, Angelo Bulpiani and some co-authors 2 years ago. So the theory, which we redid and generalized a little bit, but basically redid the theory of Neher and Schreiman, will tell you that under suitable limits and so on, the distribution of genotypes in a population follows a population which we all know very well from equilibrium statistical mechanics. It's the Ising model, or the Potts model of thermal equilibrium, where the xi's here will be either a dichotomic variable, so that would mean a 0-1 variable. That would be the case if you have only two variants and one loci, so a biallelic locus. But it could also be more, so it could be 4 if all nucleotides could be happen, or you could think of the locus as a little bit larger, and then xi could the values of xi could be the possible variants that actually are observed in this locus. So in a protein, you could have any of the 20 amino acids and a gap variable. So there in this one, you have 21 different locuses. Then you have a Potts model with 21 states. And you have single term. So this is the term here written in the exponent HIXI. That's reflects additive fitness. And then you have two body terms which reflect epistasis or synergistic interactions impacting on fitness. Yeah, so determining the such parameters from data is a problem of statistical inference, which has been studied since quite some time. It's called in statistics learning in models in an exponential family. It's sometimes also called the inverse Ising or inverse Potts problem from the physicists who work on it. And it found a very important application in predicting protein or properties of protein structures from tables of many homologous proteins. Of course, in that particular application, those methods are now overtaken completely by deep learning methods from Google, has been reported in the press. Anyway, I was involved in this actually a bit more than 10 years ago. We wrote a review, now not cited very much because in the last years, because there's a much better, much more up-to-date review written by Ricardo Zecchina and collaborators in 2017. And for the biological minded, there is this wonderful review written by Simona Cocco and her collaborators, including René Monasson and Martin White, now already 3 years ago. So there is a technique of finding the parameters of such models from data. You can just apply them. There is now packages and well-known software how to do that. And if you do that on the data which was available in GISAID up to August last year, you'll find here's just a top scoring with some filtration. I think I'm towards the end, and maybe I will tire you a little bit, but maybe it's interesting to see that many of these locuses here are things that you have seen before. So 1059 position in the gene NSP2 comes together with the 25,563 position in ORF3A. That happens to be a mutation precisely of this type appears in the South African variant. The others in this table, we didn't know that when we published the paper, so that's we were excused, I think. The others in this paper don't appear in the UK variant, the SA variant, or the India variant. And you can see that so what does it say? What does such a table say? It says that there is a statistical dependency of that the variations in this, if I look at this line 21, for instance, in position 1059 and position 18,877, they are not independent. So there's a tendency of them to be either mutated together or staying in wild type together. And it's actually a very typical thing for this kind of analysis that the very long lists, you have 30,000 positions, you have many hundreds of mutations, depending on the threshold, how you put the threshold. So by hundreds of mutations, you have tens of thousands of pairs. And still, according to the standard method, you end up with actually a rather short list of things which you will find reasonable to believe in this kind of global data analysis. So it's interesting, I think it's interesting that the viral gene ORF3A appears in several interactions. Since I'm sure that you heard a lot about spike gene and different things, maybe I'll show you something about the ORF3A gene. The structure was found by cryo-EM last year. I think it's published now, but it was the bioarchive when I did the slide. It is a protein which is basically unique to this family of coronaviruses. So there's very little variability, and you cannot predict, at least not very easily predict its structure by using the methods of AlphaFold or AlphaFold2, the famous method, now famous methods by Google based on deep learning. And it is a protein which is very much involved in serious aspects of SARS or COVID disease. It's been known already since the previous SARS epidemic that it upregulates expressions of certain. It is involved in the inflammatory response in the lungs, which is one of the most severe aspects of the disease. And well, we did, of course, we tried to see if there was some drugs already for this protein, which one could propose to use in combination with others. But for this drug, at least in the compilation, which was published last year, there was no drugs for this one. Maybe there is now. I hope so. Okay. Now, coming back to the data, why is this a kind of interesting data analysis? If you abstract a little bit from the biology here, is that these predictions, which are obtained through the global learning, so these are using theoretical arguments that predict that the distribution ought to be of the type of an exponential family, which of course is based on the certain assumptions that recombination in this case is a reasonably strong process. So then you believe, then a prediction of this is that the parameters of those models in exponential families are certain functions which you can find in our papers, and you can go further back. And also, we have some theory papers checking this in other models later. So you can translate models of evolution with certain parameters describing the strength of natural selection, mutation, and recombination, and so on. You can translate those model parameters of the dynamics of the whole population into parameters describing the stationary state of the population of this exponential type, or if you prefer, the Asymptotes type. And you find that these parameters are stable, much more stable when you add more data. This is the data which we published a year ago, which had then stratification of the data up to August last year, and we compared the dashed lines, which are these parameters from of the models in exponential family, to the most correlated pairs. So that's of course a very simple statistic to compute, and which is also a measure of statistical dependency, but it is not a more an equally concise measure of statistical dependency if you have some a priori reason to believe that the full distribution is of a certain type, in this case the exponential type. And you can see that the rank of the most, what started to be most correlated in the beginning went down eventually quite a lot, while then they are unstable in the rank. That's these kind of lines going downwards to the right, these ones here, while the ranks of the of these terms in the exponential model are much more stable. And this observation was stable until December last year. That's the latest data which have done, which we haven't written up yet, but which are on this type. So the moral of this story is that, of the second story, is that the generative models, in this case of the exponential models, are more stable than correlations as a measure of statistical dependency in the GISAID data. And this, I think, should be an encouragement to try many other kinds of data analysis of this fantastically rich data. But it is in particular consistent with recombination having an effect, not only in some certified examples, of which I gave some pointers to the literature, but also on the global viral population and its the population structure. And that has, of course, implications to how much you can believe in reconstructed phylogenetic trees from data. And so, of course, also it should be a reminder that there's more to SARS-CoV-2 than spike. Now, of course, we have now vaccines against SARS-CoV-2, which we didn't have a year ago, and those are mainly against spike. So still, spike is, of course, important, but it may have been, it could have been, it wasn't expected that the vaccines would be available in 1 year. Usually it said 3 years. Maybe in some future pathogens, it will be treatment medicine against the infection, which is more useful than the vaccines. Who knows? Anyway, there's more to the biology than just spike. And with this, I think I am done, and I think I am more or less on time. This is a beautiful visualization of the kind of couplings that you get with all the different genes written out on the rim. So if you look at the slides will be deposited, you can compare where all these different points are. Thanks out to Hongli Zhang, who I guess is in the audience, Vito Dicchio, Edwin Rodriguez Horta, who did this his part of the work from Cuba actually, and Kajsa Torell, who was at the time in Stockholm and then moved to Göteborg in Sweden, and Joel Yu and Richard Nordén, who's also in Göteborg. And I should also thank Martin White in Paris, with whom I've been discussing these issues off and on for a long time. Thanks a lot.
Okay, thank you for a very nice talk. We would all clap if we were listening to this person. So with the questions, there's 2 ways of asking questions. One is in the question and answer box, and I can also unmute you so you can ask questions in person, and you can also put your hands up. So we'll wait a few moments.
I see that Hongli Zhang is also a panelist, so she can also say hello to everybody.
Yeah, I add her as
hello, everybody.
Hi.
Hi.
Can you hear me?
Yes, very well.
Yes,
okay.
hi.
Hi.
Yeah, so Professor Seng was in a sabbatical with me in Stockholm, actually during the pandemic. So we got a lot of work done. And now she's back since about a year in China.
Yeah.
Okay, I'm very happy to take any question. Yeah. Mm-hmm.
I wonder, so I may just kick off with a question. I wonder, so, I mean, there's a lot of these academic terms in your talk, which I really cannot quite understand. But I mean, this is really one of the most difficult talk I have heard. But I really want to ask, in South Africa, we see spikes of these COVID-19 infection, and also these are driven by different mutations, etc. So do you see such periodicity in your data somewhere that may predict some more variation in the future, which could even just make things go over and over again?
Well, okay, we didn't look for it, and I doubt that we can. We don't have that data up to now. There is, and I don't think that has been done on SARS-CoV-2, but there is a lot of work in Paris by Martin White and others on other pathogens, mainly bacterial pathogens, trying to trace out the fitness landscape. So predict the fitness of a variant from this kind of analysis, which I did which I presented in the second half of the talk. Of course, it depends on the precise on the data and on the bacteria, on the biology of the organism, if it's appropriate or not. At least that's my point of view. But there have been some very good results in that direction, so that you have the variability of the organism, and you predict from this the fitness or lack of fitness of variants which have not yet been observed. But this is for other organisms. But it's an interesting problem. Sooner or later, the virus will try all kinds of combinations that's possible. And so if one could find in advance that there is one which should be very dangerous, one could scan for it and say this is a danger thing, or maybe one could even say it's not for now, not for this pandemic, but for another pandemic. One can surely imagine that there could be a program of if not creating the vaccines, at least creating the templates to quickly be able to create the vaccines for something which hasn't yet been found. Yeah. That I would say is more like in the background of a motivating idea of this whole kind of research. Of course, there are many things which are interesting, but down the line, eventually, of course, it would be nice to be able to say that this kind of organism now expanding into a host where it was never before, which in this case is the virus SARS-CoV-2 expanded into human, it could acquire more dangerous mutations, and there could be these and these and these. Yep.
Okay, we have a question from Hercules Combrink. So let's see, I will unmute you. So maybe you can ask your question now.
Thank you so much, Professor. And thank you so much, Professor, for the talk. I think it was incredibly important. As a biological scientist, went data science, I do have an absolute appreciation for the complexity of the work that you worked for. Professor, I actually have a more of a fundamental question. So I know that the absolute focus of it was specific to the variants that were proposed, etc., which is incredibly important. And I think the work that you did is incredibly vital, especially the critique on it. One has to be critical on this. My fundamental question for you, Professor, is, have you at the back of your mind thought about a fundamental mechanism that can be linked to some kind of epigenetic control mechanism that goes beyond the genetic sequences that you see, to try and identify a pattern or a system or a mechanism for why mutation variations take place? So it's a bit more of a fundamental question of whether you are able to see trends in the information that can point to a secondary or a tertiary mechanism that can explain potentially the rate of mutations that we are seeing. And not necessarily for SARS-CoV-2, but as a fundamental concept within biology. Thank you.
Okay, let me unpack the question. So epi means in Greek kind of on the surface or adding, and epistasis and epigenetics both share the epi, but they are different concepts. So let's first clarify them. Okay, so epistasis is not such a fantastically common word, but it's a well-defined epistasis or epistatic interaction. And this describes in biology the fitness effect of mutation of pairs or larger set of mutations. That means changes in the genetic code, which are not additive to the separate changes that you find. And those are the ones that we looked at looked for in the second part of the talk. Epigenetics is about ways that that organisms can inherit properties without changing the genetic code. It was for, let's say, quite some time ago, I would say this was considered to be a heretic notion. Everything came from the genome. That was the central dogma. But I think it's well accepted. There are journals and everything about the epigenetics, and there are such effects. There are many different kinds of epigenetic effects. I personally have only worked on one, which is almost so simple that the biologists don't really accept it as epigenetics, which is the stability of the gene expression in bacteria, that means that you have a gene can be expressed in this case, two different ways, or you have two genes that mutually suppress each other. So either one gene is expressed or another gene is expressed. And this state in E. coli can be inherited for hundreds of thousands or millions or even more generations. So it's stable on the same time scale as the underlying genetic code in E. coli, which basically you have one mutation per base per generation, and at least by rule of thumb. It used to be that one. And so there you can it's an effect of the stability of control circuits, which you can even write down. There are very classical examples, which the biochemists, long before I got into that business, had figured out most of, well, of course, the components, but also many of the kinetic parameters, and you can really verify this. So that is 20 years ago for me, and the science is basically 40 years old. Now, all the other types of epigenetics, I'm not really, a person to answer to this. I am not aware of any epigenetic effects in viruses, but that might be that I am myself not. It could be that it exists and I never heard of it. I think simply because the virus is just a small particle and it's packed. It's unclear what else is there that could, in the virus, prolong the memory from one generation to the next. But biologists maybe know other things. Maybe there are some viruses which have something like that. Yeah, I guess that's as good an answer as I can do.
Okay, any other questions?
There is a question in the chat from Zurab Jalidze, and it's, do vaccine encourage more or less mutations to the virus? Are there ways to measure this with current data, or it's too soon?
It's a very good question. Theoretically, one would expect that the vaccine changes the fitness landscape, so it doesn't encourage the individual disposition of a mutation to occur in one organism, one virus, but it certainly tilts the game, so that the chance of one mutation going up to a large frequency and eventually reaching fixation, that's changed because the first you have the whole fitness landscape is changed if everybody is vaccinated, but it's also changed if only part of the population is vaccinated, because then you have a more variable fitness landscape in which the virus evolves. I think that's something that we should be able to look at with the data, because the time stamps are known when all the data, all these GISAID sequences are taken, and the vaccination frequency in the different and which vaccines were used in different countries are known. It's a question of data fusion and manpower. It could be done, but I don't think right now we don't have the resources to do it, because one would have to. It's not a question of working one database. It's a question of bringing together data from different databases. I think it's a very good question, and I hope that if not we, then somebody else could work on it. Yeah.
Okay, thank you. And I think the person who asked the question is satisfied with the answer.
It sounds, yeah, so
thank you. Yes.
Yeah.
Let me ask a question. With the second part of your talk with correlated effects, to what extent can you, take into account, evaluate statistical significance and sort of take into account sort of noise correlations? I didn't quite understand that.
Okay, it's we did quite a few tests which are in the paper, and I had one slide that's a backup slide, but I guess it's too late. I will not go back to it now, but you have the figure in the paper. So we did a randomization test checking if one could explain the effects by phylogenetics. So that is like you have you're comparing to a null model, but this null model is not accessible through a formula. It's accessible through sampling many times. So it's a bi-Monte Carlo procedure. And that was technically the most difficult part of that paper, and the most novel one. And it was actually contributed by Martin White, your colleague in Paris, if you don't know him, but he has extremely high standards. So he said, I didn't do enough, Erik. My postdoc can sign the paper. So that's why they did it together. But there is also a paper when they report the results on other data, not on SARS data, but this kind of things. So that's what we have done. Okay. The question of assessing statistical significance in these kind of global methods is not trivial. It's a part of advanced statistics, and you find a whole literature if you dig into the methodological literature, where you have the problems would be of the following type. Given a number of samples, n samples, generated according to one of these models, what fraction of the strongest statistical interactions as assessed by those parameters in those models will you be able to retrieve? Okay, and the literature on this thing must be surely hundreds of papers, but at least I can quote to 10 if I would have to. And it's not trivial, it depends. Sometimes it doesn't work, sometimes it does work. And it depends that you must make assumptions in all cases on the underlying distribution. And there is a difference of particularly it's different if your goal is to retrieve the largest terms, which is what I use always. And if you have a more, let's say, mathematical goal of saying, I want to retrieve all non-zero terms. That means I want to retrieve all the effects, all the underlying Markov, the graph structure of the dependencies I want to see, and what's my chances of finding it, say 100 loci interacting and so on. So if you go that second route, then chances are usually zero, but in the first part they are in many cases quite high. But it's not so that you can give a very classical answer. It's not that this has been done by the statisticians 50 years ago. It's an ongoing literature which is continuously published. from both, I would say, physics applications and also from rigorous mathematics and statistics.
Okay, thank you. Any more questions? Okay, well, thank you again for a very nice and inspiring talk and taking your time to be with us. And thanks to the audience too for participating.
Thanks, it's my pleasure. Thanks a lot. Bye.
Thank you very much, Erik.
Bye.
Bye.
Thank you. Bye-bye.
Bye. Bye, Ongli. Nice to see you again.
Bye.